So this portion of the presentation kind of lines out for you different viruses and how they impact um, the humanity, but how they impact individuals, but also how they impact groups, and why we care about isolating for periods of time and that sort of thing, and the severity of the viruses, because a majority of the pneumonias that occur are a result of viruses. So viruses, as I've got a note up there at the top of the screen, most common of all human diseases, um, just because of their capacity to be transmittable and their potential for mutation. As you guys are probably aware of after hearing all the news related to COVID-19 and that sort of thing. So we'll touch on that just a little bit. So what I want to point out to you here is on the left-hand side, under the very, as you look at the screen, the very first column there are a few areas I want to identify for you and kind of point out what these things really mean. So presentation, what I mean by this is how it characteristically shows up, and that's what's going to be delineated in that row. Strains will identify whether, as example, in rhinovirus there are multiple, many strains, too numerous to list. And then you have various strains of coronavirus, and then within that, there are mutations within a strain. That, as you well know, there are multiple mutations of COVID-19, and I'll discuss what that means as far as strain goes. Then transmission, how this particular virus gets propagated from human to human, the incubation period, which is how long it is before this person is going to manifest symptoms. And that becomes important, as we're going to talk about in just a moment. And then receptor or vectors. So by receptor, what I mean is if there are receptors with which these viruses interact to propagate what they are. And this particular area is not so much about the receptors, but where they're located, which has a direct bearing on the severity of that particular viral illness. And then shedding is the length of time that individuals are, once they are contaminated with the virus, how long they're shedding that virus in a way that they can be infectious to other people. And then immunity, whether they actually sustain immunity to that particular virus or not. So to begin with, we have rhinovirus here. This is the common cold. So if you have a cold, runny nose, don't feel well for a couple of days, and you're congested in your head, you have a lot of sinus or allergy type symptoms, that's because of rhinovirus. Now, if you notice here, its transmission is by contact transmission. So that means that you have to come in contact with my secretions in some way, whether it's I sneeze in your face or I sneeze into my hand and we shake hands, whatever that might be. And in a period of time where the virus is still alive. So there's some of these viruses are not sustainable for long periods of time. By long periods of time, I mean 24, 48 hours, whatever, on a surface. They just don't live that long. As opposed to Acinetobacter, which can live for multiple days on a dry surface. Now, this next piece about rhinovirus is distinctive because you notice here the receptors with which it interacts to cause the response, the symptoms that you manifest, are in the nasal mucosa. So what this means is rhinovirus doesn't characteristically end up, rhinovirus itself, creating a pneumonia for someone. So I put it here as a contrast because it's limited in its manifestation, because it's limited in the areas in which it affects cells to propagate. And once you're exposed to rhinovirus, characteristically, you sustain immunity to that particular virus. Now, coronavirus is a virus that we're now familiar with because it's associated with COVID-19. COVID-19 was one of 7 strains originally that existed for coronavirus. Coronavirus was also Its earlier strains were also responsible for the common cold. So rhinovirus and coronavirus were two viruses that produced common cold. Now, in the latter strains, such as SARS, MERS, and COVID-19, that it could present as a cold, or it could also present as, it could evolve to pneumonia. So the difference here is that one, it's by droplet propagation. So if you're projecting your voice and just the moisture from the droplets in your voice, which you can't even perceive as you're talking. And as I'm talking right now, I have no moisture that I can perceive that's moving through the air, but droplets in that projected voice could then transmit this virus to other people. So that makes it a little more transmittable. It also has a longer incubation period. So you notice here, 1 to 14 days. So you could get coronavirus, one of these variations of the coronavirus, and now thinking about COVID-19, since it's so endemic in our population, you could get that in one day. You could start having symptoms, or you could not have symptoms for many days, 10 days, 11 days, 12 days, and then you start having symptoms. Or you could potentially not even have symptoms at all. And in that time period for 14 days, this thing is brewing and cultivating. And notice, if you look down, notice the shed time, 10 days. So you have a long period of time here before you're ever aware, if you become aware you have symptoms. You have a long period of time in which you're shedding this virus and potentially infective, whether you are aware you have it or not. This is what made coronavirus, COVID-19, so virulent, and and how it turned into such a pandemic, because people would not be aware of symptoms. People were contagious for a long time. People were shedding the virus long after their symptoms subsided, not aware that they're still shedding. And small droplet and large droplet propagation is an easy way to transmit this. Now, the reason it became so virulent just refers to its capacity to be spread and cause infection in a broad range. But it also has a flavor of its lethality, which is a slightly different component. So the severity of the illness, despite its virulence, the severity of it was related to this section down here where it says receptor and vector, because the receptors were in the mucosa, from the nasal mucosa to the tracheal mucosa to the lung parenchyma, or the lower lung fields. So all of those lung fields had the potential for propagating and propagating the virus and worsening the symptoms, which means if you can get virus in a trachea, in a bronchus, in a alveolus, and it propagate, now you have a more severe lung infection and manifestation, and a greater severity of manifestation, particularly in the context in which we didn't have a means to treat this initially. So that's, as you can see it right next to rhinovirus. Again, two viruses that can cause the common cold, but the rhinovirus, 1 to 2 days before people start noticing symptoms, and they shed for a minimal amount of time, and you have to get in contact with their secretions, as opposed to coronavirus, which is, you may be 14 days before you show up with any symptoms, 10 days that you're shedding, you can get into the lungs, easier transmission through droplet propagation. And notice also that the immunity to coronavirus was not sustained and still is not. So you can have you can have coronavirus and then recover from it, and after a period of time as short as weeks, you can turn around and be infected again and have an illness from it again. So this is a challenge in maintaining immunity or preventing the spread. As you can see, we've talked about containment. Now notice how coronavirus compares with influenza. Influenza also manifests as a common cold. However, it can propagate to pneumonia, because if you notice that it affects, if you go down and notice the receptor and vector, ciliated columnar cells in the nasal mucosa as well as the bronchi. So influenza can propagate to pneumonia. However, notice the thing at the very bottom. Once you encounter influenza, you maintain seasonally immunity to that strain. So what that means is, typically for the winter months, influenza propagates typically in late September, October kind of time frame, and people encounter it through the winter. And then in the spring, we don't have as many influenza cases. That's not because of any characteristic of the virus in warm weather. It is because of behavior. What I mean by that is human beings in the October months in our country and in the northern hemisphere in September, October, November, things start cooling off. People start spending more time inside. So they propagate this among themselves through the winter months. And then when they start moving outside again, they tend to transmit the influenza virus among themselves. So cases dissipate. So for those reasons, we're able to sustain seasonal immunity. Our body sustains seasonal immunity, and that protects us from having recurrences multiple times within the same year. So you can see how these different components affect the propagation of these among and between people. Notice also that the incubation period, 18 hours to 3 days, so a relatively short amount of time that you start showing up with symptoms, and a very much shorter period of time, half or less time, that you're shedding with the virus. So this makes influenza much less transmittable and much less likely to produce an epidemic. And then you have adenovirus, which also can manifest as common cold, but can propagate to pneumonia. It is most common in infants and children. There are 7 subgroups here and 51 serotypes. And generally, transmission is through the conjunctiva. So, people, kids encountering the bacteria somewhere, whether it's, from touching body parts, and then their butt, and then hands going to mouth or eyes, that can transmit it, as well as inhalation. And here you notice that this propagates through cell lysis, which is the process in lymphoid cells, the process of cells dying and then opening up. So that's how it propagates from one cell to the next. So you can see that the method of propagation is different. The manifestation, you can see the incubation period, 2 days to 2 weeks, and 5 to 6 days typically after exposure, but up to 2 weeks. And they shed for weeks. So this is a very potentially communicable adenovirus. So this is why we isolate kids when we suspect symptoms of something like this. But notice the immunity to this is sustained, unless, of course, in groups that have, that are immunocompromised or something like that. That might be an issue. But you notice that they sustain immunity, which means they're not going to get multiple manifestations of adenovirus because they'll develop immunity to it. Enterovirus is very infrequent as far as producing the common cold. The incubation period is about 2 to 10 days, and notice it's shed in the feces. So this makes it much less communicable, transmittable, because this is enterovirus is one that can show up in adults, but as adults, as a general rule, we don't play with our poop as much as, kids, infants, small children just don't have that discretion yet, as you may or may not know if you've had kids. The enterovirus, its symptoms manifest in the intestinal mucosa. So while it can produce pneumonia, it's very infrequent because the usual manifestation here is in the intestines, is where you see the manifestation of enterovirus. Now, the last thing I want to touch on here is briefly, because you're not going to be tested specifically on this particular point, but it is good to be just kind of familiar with what is meant by variant and strain and mutation. So you see, here are those words tossed around on the news and articles among providers, and to be kind of clear about what is meant, a mutation is a change in a virus or any organism really, but in this case a virus, a change in a virus's DNA that makes it distinguishable from an earlier version of that virus. So they look very similar. So if you think about a human, a mutation would be, like if you think about maybe perhaps twins, but one of them has a different hair color. So that would be a mutation, that, or different eye color. That's mutation is a different gene expression. So in this case, that mutation is enough of a change in that strain or that virus's DNA that it can be distinguishable from another parent version of that virus. Now, the key here is that when you have a mutation, that could be one gene or it could be a couple of genes. The thing is, it responds similarly to treatment. In other words, so think about like an antiviral drug. So if you have a virus, it responds to an antiviral drug, it mutates, and it has a couple of different genes, but it still is treatable by that same antiviral drug. That is a mutation. A strain is when you get multiple genes that altered, multiple mutations in that virus, yet it still responds to that conventional treatment. When you have that's a variant. When you have a new strain is when there are enough mutations that produce a change in that virus such that it doesn't respond to previous treatment. So let me kind of summarize that briefly. A mutation is a change in one or two genes that make the virus distinguishable from its parent. A variant is when you have multiple mutations that make that virus different from its parents. And a strain is when you have a new variant that responds differently or does not respond to previous treatments. So a mutation responds to the same antiviral as the parent. A variant responds to the same antiviral as the parent. A strain does not respond to that and may need another pharmacologic agent or another treatment method. So that's what's meant by strains versus variants versus mutations. So just wanted to clarify that. This picture right here is meant to kind of give you an idea, conceptualization of how viral infection works versus bacterial infection. And there are a lot of misconceptions, and we'll discuss this in class, about what viruses do and what immunizations do related to our DNA. We'll discuss that in class. But the key is that a virus inserts itself into the machinery of a cell and uses the DNA replication machinery to produce multiple copies of itself. Bacteria use the environment of the cell to propagate itself. So it self-propagates, not using necessarily the machinery of the cell, but creates different signals. As you can see here, a signaling cascade and responses of integrin and different inflammatory mediators, similar to and some overlap but different from the viral inflammatory mediators. So the bacterium does not insert itself into the cell to use the cell's machinery to replicate. It grows in an area, whereas the virus, as you can see in this image, inserts itself into the cell. Currently, there are, I want you to be familiar with these vaccines. We've mentioned before Prevnar. The previous slide said 13, and actually the newest information is there's actually 20 strains of streptococcal bacteria that are covered by Prevnar. Pneumovax covers 23. I don't have the detailed overlap, see what overlap there is between the streptococcal bacteria that are covered between those two. Then the flu virus is an inactivated virus. So with the flu vaccination, you have the virus, but it is basically the shell of the virus. There are no replicative guts to it. There's no machinery of the virus to produce the infection. People may have symptoms that are flu-like symptoms from having had the flu vaccine. They did not contract the flu. What they're having is an interferon response in response to the vaccine, which is what you want the vaccine to do, because that means that your body has recognized the vaccine and is responding to it as if it were the virus and developing your immunity. So some people can feel really crappy from having a flu vaccine and the COVID-19 vaccine as well, because it activates the viral immunity within our system. So that can actually be mean good things. The COVID-19 virus uses messenger RNA to reproduce a viral antiviral response to produce the immune response. Now, what is unique about this is the COVID-19 vaccine only has a very small portion of the DNA that is part of the virus. What they did with the COVID-19 virus or vaccine is they isolated the portion of the DNA of the COVID-19 virus, the portion of the DNA that produces the spikes that you've seen pictures of. And they were able to isolate that it is the spikes that is the area where the virus attaches and subsequently propagates. So we were able to take that portion of the DNA and create a vaccine that would identify those spikes on the COVID virus and develop immunity to the spikes. So it is a very small portion of the COVID DNA that is in the COVID vaccine. So it's a very, very safe as far as vaccines itself, it's very, very safe in not causing the illness that it is meant to treat. There are some antiviral medications out there that can be used to treat viral pneumonias. You will see things like Tamiflu, and which is oseltamivir, and then peramivir, and those two drugs are used to Tamiflu and Rapivab. Those two are used to mitigate the symptoms and help expedite recovery. They don't prevent the virus itself. Now, amantadine, as I've listed here, helps prevent viral infections from influenza. But again, the key there is you have to use it when you are not having influenza and you're going to be perhaps in areas where it may be. You've got to be taking it ahead of time. You will also see drugs like ribavirin and rimantadine and zanamivir, which are also used in the treatment of influenza and managing viral infections. So those are some drugs you may see out there from time to time to combat this. With influenza, it is not uncommon if someone has basically an ambulatory case. So they don't have multiple comorbidities, such as immune compromise, diabetes, etc. Then they may be managed by simply letting the virus run its course. If they are propagating, if this is extending into and developing pneumonia, then that's where you'll begin to see these medications used. Or if there's a risk that they may develop pneumonia because of their immune compromise, then they would use these drugs to help prevent a severe manifestation. COVID-19 has a very limited treatment pharmacology, so there aren't a great deal of drugs out there. Remdesivir is the only antiviral that's approved in the treatment of COVID-19, and that says PDA, that actually I need to fix that. So it says FDA. It's the only one approved by the FDA for the treatment of COVID-19. What's noteworthy about remdesivir is it essentially shortens the course of the viral infection. It doesn't combat the virus or decrease morbidity or mortality, but it has shown promise in at least mitigating some of the mitigating the length and duration, which may indirectly contribute to reducing morbidity and mortality. There's another medication, a monoclonal antibody there, batelovimab, which blocks ACE2 the ACE2 receptor and the coronavirus spike interaction. So one of these receptors that coronavirus interacts with are the angiotensin converting enzyme inhibitors, angiotensin converting enzyme receptors. And that's where the spike interacts. And this medication, this monoclonal antibody, blocks that receptor and prevents the spike from interacting there, which prevents the coronavirus's propagation. And then also there are two other monoclonal antibodies there that they're not antivirals, they're monoclonal antibodies, which basically what we're doing with these two medications is we're giving the patients antibodies to COVID-19. So what happens in response to contracting COVID-19 is your body responds by developing antibodies to that virus. And this is why using covalent plasma in the early days of treating COVID-19 was effective, because essentially what we were doing is we were giving patients antibodies from another patient to help prevent the illness or enable recovery, help their bodies fight that infection. They, through research, were able to analyze and isolate those antibodies and then subsequently reproduce them. That's what's meant by monoclonal. So they were, through a cloning process, able to reproduce those antibodies in a way that could be administered to another individual and not be dependent on extracting someone's plasma. So that's what the monoclonal antibodies are. We're essentially giving someone antibodies to fight the infection. So they're not antivirals. They are antibodies that help block the propagation of the virus in that case. So we will pause here, and in the next video, we will talk specifically about some issues related to COVID-19 and other components of that viral infection.